Hello, everyone. After a break, we are excited to resume CMSA Colloquium series. Today, our speaker is Evelyn Tang. Dr. Tang is based at the Max Planck Institute of Dynamics and Self-Organization, where she is leading a group at the Department of Living Matter Physics. Her group investigates the emergence of such phenomena as cognition and brain development, for example, how the topology of white matter changes across development, or the geometry of neural activity during effective learning. Prior to MPI, Evelyn was a postdoctoral scholar at the University of Pennsylvania in the group of Dani Bassett, where she focused on network dynamics and controllability, brain development, and effective learning. Dr. Evelyn Tang received her PhD in physics at the MIT, where she worked in condensed matter theory with Professor Xiao Gang Wen. She was also a Gates Scholar at Cambridge University. Evelyn, we are excited to have you. The floor is yours.
Well, thank you, Sergey. That was a very comprehensive introduction. So thank you very much. And your brain network looks and lights up in a very different way from my brain. And yet we can communicate and maintain many similar biological functions. Or if we go down to the smaller scale of cells, there's often a lot of stochasticity and small copy numbers. And yet living systems show robust function despite all this heterogeneity and stochasticity. For example, we see persistent timescales in circadian rhythms or in memory. And of course, developing a mathematical framework to answer these questions is a big task, but we normally have the framework that we have to average over many particles usually assumes identical particles in equilibrium and isolated from the environment. And so that's one reason why I'm very excited to speak to you today. This is such a great audience with backgrounds from so many different disciplines, which is something that we'll need to tackle such a big question. So I'm going to show you today my little step towards answering this question. And this is a sneak preview of what we'll be showing, which is that we propose models that support dynamics that are reminiscent of this emergent behavior. And these models actually all begin with the edge state of a topological phase, which I'll explain in more detail. But just to give a quick overview, we have a large molecular reaction space, and it turns out that in these systems, the system will move along the edge of this molecular reaction space to form a global clock, an oscillator. We'll see that we're changing just one parameter of the system. Then it's possible to see behavior that looks like stochastic growth and shrinkage. Is it possible to mute that sound or not really? I'm just curious. Or is it just on my end?
Well, it's I'm not sure we can mute it, but okay, it's not recorded.
Okay.
Sorry about that.
And changing just one parameter in these global cycles that emerge can lead us to see stochastic growth and shrinkage, which is reminiscent of the dynamical instability of a microtubule. And then taking two of these oscillators and since that they are now, we can now couple them by just allowing their reactions to depend on the same pool of reactants. So without having to directly couple them, but just using the same physical constraints, we'll see that we actually obtain emergent synchronization or anti-synchronization. So this is just a preview of what we'll be showing. And I want to go back to the star of the show, which is the edge state of a topological phase. And I'm going to illustrate a topological phase by using the canonical example of the quantum Hall effect. And this is a two-dimensional electron gas where in the presence of an external magnetic field, the electrons just make loops, these are cyclotron orbits. And so in the bulk of the system, they don't go anywhere. As you can see from this semi-classical cartoon, they just stay in their space and make little circles. But at the edge of the system, what happens is that they actually hit the edge of the system and they sort of run along to form these chiral edge currents, one in this direction and one in the opposite direction. And what is nice, you can see now is that even if, let's say, the edge had a hole in it, or if there was some blockage, that this state would just simply move around the boundary and keep going. And this is a form of robustness that allows them to continue to persist, even if the system was not perfect. And while this has been a lot of fun in quantum systems, in the past decade, there have also been proposals for these states in classical systems. And in fact, there have also been states for these, for topological models in stochastic systems, in particular in these one-dimensional systems, where here you can also see this idea of a bulk and an interface. And here now there's winding in this bulk and different windings on the left and on the right that leads to probability density building up at the interface. And in these 1D systems, the edge is 0-dimensional, and so the edge state is not going anywhere in these systems. This is simply a polarization. And so our question was, can we obtain propagating edge transport like in the quantum Hall states? So if we want to think about stochastic systems and in particular biological systems, of course, we don't have, it's not an electron gas. And so one would think, well, how would this even come about? And we actually identified, there are many ingredients that could contribute towards this. And one of this is the fact that living systems exhibit many out of equilibrium transitions due to the consumption of fuels such as ATP and GTP. And in fact, we've identified futile cycles as being able to play the role that bulk electrons play in these systems. And they are called futile cycles because they consume energy and they seem to be moving in some kind of perpetual cycle. And this takes a lot of energy, but yet they don't seem to have any apparent function. They leave the system apparently unchanged, and they appear in many different biological systems. And so these could play the similar role of electrons in the bulk of the quantum Hall gas that sort of go around and don't seem to do anything, but their presence supports chiral edge states. And this could support a powerful mechanism in biology in which structure determines function, such that we get emergent global cycles that are governed by the physical constraints of the system, which in this case will be a chemical reaction space, rather than having to fine-tune microscopic timescales, such as to obtain the kind of global timescale that we want. I just see that my Xiaogang, my former advisor, just entered the room. This is clearly out of equilibrium physics because we see that these are directed cycles, and this gives rise to actually very pretty mathematical properties. In particular, here these stochastic systems are described by the master equation, which you can see here, and the physics is governed by this transition matrix. Now, this transition matrix is non-Hermitian because these out of equilibrium or directed transitions will make W non-Hermitian. And this is mathematically has been of great interest in the past, again, few years, because most of our topological theories are mostly developed for energy conserving closed equilibrium systems. In other words, for Hermitian Hamiltonians. And yet we know that living systems generically will break all of these conditions. For example, directed interactions are very common in biology, for example, in predator-prey interactions. And what is really nice, and this opens up a whole sandbox of pretty things. And I saw that Juven raised his hand. Is that something that?
Yes, Evelyn, may I ask? Very naive.
Oh no, please.
So the question is, you mentioned this the age state. I suppose like 1 + 1D age state, maybe on the boundary on 2 + 1D cell. So my question is just that whether the dimensionality of this cell to be 2 + 1 or 2-dimensional space is somehow crucial and important, because you mentioned the chirality, chiral. So
absolutely.
I'm lack of knowledge here, so maybe you can make a comment why this 1 + 1 and 2 + 1 is important, perhaps from the biological point of view.
Absolutely, yes, and as I mentioned, if we just have a 1D stochastic system, as in these proposals from the Chicago group, we're going to get edge states that don't go anywhere; they'll just remain stationary. And so, to get a chiral edge state, we do need a 2-dimensional system. And so, my models, which we'll see in just 2 more slides, will be proposed in a 2-dimensional state. And this is a coarse-grained description, but it is a fairly good description because also in biology, often you get some main timescales that may be of interest to the system. And so there could be 2 particular dimensions, for example, that are relevant. And I'll show examples of this.
Okay, thank you.
And so just this is the last comments about non-Hermitian physics. And now we do get many pretty mathematical properties because now since the eigenvalues can be complex, they can not only be complex, but wind around and create interesting topological invariants, for example, in this paper by Liang Fu and friends. And we also get interesting singularities called exceptional points, where the system becomes singular here because the eigenvectors coalesce into just one eigenvector, for example. And this is distinct from Hermitian systems where you still can have degeneracies in the system, but the system that does not become singular because the system will still have distinct eigenvectors. And we get other things such as square root branch points. So this is just a brief overview of some things that we'll be seeing. And now I'm going to go into our models, and then I'll discuss the topology, and then I'll look at also what considerations of biology can tell us about these models. So let me show you our models. And this, as Juven was saying, we do look at a two-dimensional reaction space, so it can be laid out in such a form. And this is can be thought of simply, one example could be a polymer system. And in this polymer system, we have two monomers. Here is a purple monomer and a green monomer, which forms our two dimensions. And here, transitions in the monomer space could be simply the addition of a green monomer, addition of a purple monomer, removal of the green monomer, and removal of the purple monomer. And here you can see the numbers of the monomers in these brackets, so that they would form closed cycles in monomer space, like in these black arrows. And in our model, we also have internal transitions, which do not change the number of monomers, but they allow for the system to know what transitions are allowed next, or they tag what can happen next. And so, for example, in here, we see that there can be transitions between these four internal states in the system that then allow the system to know what sort of transitions can happen next. So this would be a simple 4-state system. You could also have a system with 3 internal states, where one physical incarnation could be where there is addition of a monomer, where there's phosphorylation of a monomer, which here we denote by this little purple square, and then addition of a new monomer, and then removal of a phosphorylated monomer. So this would then form a triangle in these monomer space, and we also have 3 internal states here that allow for transitions amongst themselves. And now we can so using results from topology, what we find is that when these black arrows are stronger than these gray arrows, the system, once it hits the edge, it will continue moving on the edge. And you can see this like so. Let's say we are here, the system can only move forward. If the black rates are faster than the gray rates, it will very likely move forward. Here will move forward again. Here will most likely move forward, and so on and so forth. And you can see that this becomes more robust the stronger the black is from the gray. It will round the corner and it will keep going. And this is the same with this 3-state system. And here's where now you see that a topological protection happens, that now even if there's obstructions in the system, or let's say we have missing pieces, then this edge state will just keep going around the edge and continue. So we can verify this using stochastic simulations, and we see that the system, initially, if it's at the center, it may move around, but once it hits the edge, it will stay there. As you can see from this time trace, where initially in the middle, it does a random walk, but then after a while, it starts to go on the edge, or in this 2D plot now which has time in the 3D axis. If we plot the probability density over long periods of time, then we also see that the probability accumulates on the edge. Solving the master equation allows us to see this as well. And in fact, we can see that the edge states are exponentially localized here. And this localization at the edge then allows for this edge state to be robust even in fairly small systems, which can be the case in some of these systems that we'll discuss. And here we can just verify the same thing for this 3-state system using, again, simulations and a direct solution. So I want to give an example of how this could be realized in a biochemical space. And so here we're going to model a coarse-grained version of the Chi-ABC system, which is a protein system that governs the circadian rhythm of cyanobacteria. And this system has 6 monomers, which are arranged in a hexamer like so. And each of the monomers can undergo 2 transitions, which form our 2-dimensional space, one of which is conformational change, which is here shown in this difference between this circle and this diamond, and the other one is phosphorylation, which is shown here by the addition of this yellow circle. And so, and since each monomer can then undergo each of these changes, you can imagine that this would form a 7 by 7 lattice space with all of the possible changes that a system can undergo. And then when the system is in the topological phase, then the dynamics will move around the edge because of what I was showing you. And they will only stay on the edge so that they will undergo a large loop of where all the monomers undergo conformational change, all the monomers undergo phosphorylation, and then all of them conformational change, and all of them dephosphorylation again without having intermediate changes. And interestingly, this model is what we predict is the same thing as what is suggested in this allosteric model, which is a very popular model in the literature. And in this allosteric model, they also propose that the system undergoes phosphorylation, all phosphorylation, and all conformational change, and all of them become dephosphorylated, and all of them undergo conformational change again. However, here they have to ignore or assume that these rates in the middle are small, and that there is some difference somehow between these monomers at the end, such that the transitions only happen at the end, and they don't happen in these other intermediate ones. And whereas for our system, we don't have to assume any differences between each of the monomers, but instead this large loop happens, comes out very naturally just because of the edge state properties of our system. And this is a paper with some experimental evidence that supports the stacking mechanism and says that the conformational change happens via the stacking. So those were the models. And are there any questions there before I go on to look at the underlying topology?
Question?
Yes.
Just want to make sure. I thought this model is about a two-dimensional system, yes. But the biological system is a three-dimensional system. So
why do you, so yes, this is the one point that I was going to make next. So if you think, so this is one difference between the systems that we think about in solid state. In solid state systems, the dimensions of the system are actually given by the physical real space dimensions. Whereas here in these models, the dimensions are given in chemical reaction space. So this is a configuration space. So I'm just gonna show again just our models, just so that this is super clear, because it's a great question. And I just want to make sure that this is clear before I move on. And whereas in these systems, our dimensions are not governed by physical space, but this is a chemical reaction space. So what is actually happening when I say that when the system moves from here to here is that the system is undergoing a chemical transition from here to here. And this transition could mean something like adding a monomer or removal of the monomer, or in this case, phosphorylating the monomer, which is adding a phosphoryl group onto the monomer itself. So what we're looking here is some effective reaction coordinates, and it is quite conceivable that there can be two main reactions that govern the system. So for example, in this example that I showed about this chi-ABC system, now the two dimensions again are not two real space dimensions, but one dimension is that the system can undergo conformational change, and the other one is that it can undergo phosphorylation. So here, if there are two effective changes that are happening, then we can model this as a two-dimensional system. Does that make sense?
Yeah, I see. Thanks. But in reality, there may be, as you said, there may be some self-dominate reactions that
absolutely
from other dimensions where this give some corrections.
Absolutely, yes. And in biology is very complicated. There are probably lots of different possible reactions that can happen. But then, depending on things like timescale separations, there probably will be some reactions that that dominate. And also, one can also imagine that if there are if there are reactions that don't change the topology, then we would still have this topological state. And we know this also from thinking about topological phases in quantum systems, that if you do have some noise or some perturbations, so long as they do not close the band gap or they don't drastically alter the topological phase, then the system will still be in that phase.
Thanks.
Sure, great question.
Okay.
Yes.
I also have a follow-up question related to Liu Jun's question. So as you mentioned, the dimensionality is the reaction space.
Yes.
And sorry.
June, is everything okay?
Yes, the Wi-Fi is not stable. Let me say again.
Okay.
So there's still some notion of the locality. Seems, for example, a certain, you know, the way you draw the model, it seems like the reaction only trigger between maybe two neighbors. So there should be. I just wonder, say, this is not a very precise question, but seems like there's some locality concept between, for example, like some reaction space sites.
Yes, absolutely, there is, right. And this is, you know, this is laid out in a very specific lattice model. Each, each state can only transition to, let's say, one other state. So this is definitely, you know, very particular locality. If we look at this polymer model, we can see that this would be natural because here if we're adding polymers. So this is literally adding, you know, from 0, 0 to 1, 0 to 2, 0. You know, we have to add 2 before we can go into adding another one to become 3. So if we think about adding and subtracting, there is still a physical association to this space that would make sense for it to be laid out in a lattice. That's not real space, but these can be counting the number of monomers or counting the number of reactions that would lead to a lattice structure. So that's a great point. It's not that it's a completely abstract space. It's just not real space.
In general, sorry, let me just finish. In general, I imagine that the space here may be even not integer dimension, right? Is it possible to have much more complicated, maybe fractional, some, you know, this fractal-like space in a sense in the reaction space? I just wonder, maybe from your biological system or your knowledge might tell you there's such possibility. This is just an opening question.
I agree. This would be great to explore. Here, I would say this is the simplest possible model that we have in dimensions, but it would be great to discuss.
No problem. So it's possible in biological system there's such possibility.
Yes, biology is very complicated, and towards the end of my talk, I will talk about how thinking about biology already starts to make this to open up a lot of interesting possibilities. And
no problem. Thank you. That's great. Thanks very much.
Sure.
Sorry, so I have a question too about you mean the configuration, the chemical reaction configuration space. So from this graph, are we just following one reaction trajectories, or we have many possibilities?
So in general, there are many possibilities, but what is the nice thing about topology is that in the topological state, which is when the black arrows are stronger than the gray arrows, then the system will stay on the edge. So when that is not true, when let's say gray is larger than black, or when they are about the same, the system will just have a random walk in the middle of the space, and then the system can go anywhere. Whereas what happens in topology, and this is why topology is so exciting, is that in the topological state, then the dynamics are dominated by what's happening on the edge, and then the system will follow a prescribed trajectory on the edge.
Yeah. Okay, thank you.
Sure.
Sorry, maybe just to make sure one technical point, just on this slide. So you have 4 internal states.
Yes.
Then I think from the master equation, you will have these 4 internal states, and then you can map it to a non-Hermitian Hamiltonian that is 4 by 4.
Exactly.
Then how do you get a thermodynamic system? Is this 4 by 4 Hamiltonian somehow like the 4 block bands, something like that?
Yes. Yes.
Oh, I see.
So there will be flow of block bands, and what Liu Jin was saying is that we can look for the eigenvalues in Fourier space, but at the same time, these ideas will work even for much smaller systems because of what I was saying, that we find that the edge state is localized. So even when we're not in a thermodynamic system, even if it's a small system, that because the edge stays very much it doesn't penetrate into the bulk, so then the edge states will still be robust even in small systems.
Mm-hmm. Okay, thanks.
Great. So now we can go and look at this 4x4 matrix that Liu Jun was saying. And so here I'm going to talk about the topology of the system, which is easiest to be seen via looking at the band structure, which is this, which is what Liu Jun was pointing out. And in here, what we can see this by analyzing W, which can be mapped into a Hamiltonian. And here, what we can actually, besides mapping this to a Hamiltonian, because you can see this is very similar to a Schrodinger's equation, we can actually further calculate the Berry connection of W. And we can do that because of the lattice description that we have, which then allows us to go into Fourier space. And this link was first formulated by Zach very nicely for electrons, in which he showed how the Berry connection of a particle moving in a lattice could be calculated. And in fact, that this Berry connection could be quantized. And here, this point has already been made that typically in solid state systems and electronic systems, the lattice coordinates are in real space, but here we're looking at a configuration or reaction space. So more explicitly regarding the Berry connection, it is calculated from the Hamiltonian, and in particular, the eigenvectors of the Hamiltonian, so that we can look at its winding. And so here we can look at how the eigenvector of the system, let's say, winds in this Brillouin zone. And by integrating over the Brillouin zone, then we can obtain if we obtain pi, then the system is in the Zach phase, named after Zach, since he made this observation, which shows that the system has a non-zero polarization. And then because this system is identical to the master equation that I just showed up to Ni, so then if we do map the transition matrix W into a corresponding Hamiltonian, then the eigenvalues will have a different factor, but the eigenvectors will remain the same. And since the Berry connection is calculated only from the eigenvectors, therefore we can calculate the Berry connection of W also directly from its eigenvectors. And we can make further progress by decomposing W. And this can be typically done for this transition matrix of a master equation, where there's two parts where A looks at the transition rates from between two states, and D is the diagonal matrix. So A is the incoming flux and D is the outgoing flux, respectively. And in fact, A is what we are very familiar with in solid state physics, because it is typically written has a very similar form to the hopping for electronic systems. And in here, what we can see actually is that the A that we propose in our systems is actually a 2D generalization or a 2D non-Hermitian generalization for a very well-known system, polyacetylene, which is shown here and was first proposed in this paper a few decades ago. And in here, what you can see also is that in this one-dimensional model, there are two types of bonds here shown as double bonds and single bonds, respectively. And these two different kinds of bonds correspond to these black and gray bonds or black and gray transitions that I was showing you in our models. And in this SSH model, they show that this model contains a topological state. And so which here you can, in the regime that I mentioned, which is, or in this particular regime of this double bonds being stronger than these single bonds. And this will give rise to an edge state, which you can see here at this domain wall. And this decomposition is nice because we do know quite a bit about these this hopping matrices from quantum Hamiltonians. And in particular, in periodic boundary conditions, as Eugene was alluding to, then this can help to simplify the system a lot, because now then in Fourier space, D is just proportional to the identity, so that W and A will have identical eigenvectors and hence the same Berry connection. And so here, now we can use results for the SSH model, which describes the Berry connection of A. And we know that, in fact, that besides in 1D, which was the model I showed you on the previous slide, that the topological transition has also been studied in the 2D Hermitian model, which you can see here. Again, here you can see these two different kinds of bonds. Here now one is shaded dark and one is just a line. And here the transition in this system is at the same value as I showed you in the previous 1D model, where the transition happens with equal values of these two bonds. And this is the Hermitian model, so that the transitions or the hopping are the same in both directions, whereas in our system is explicitly directed from one to the other. And so the question would be, well, does this topological transition still hold in our system, where the system is very explicitly non-Hermitian? I see that there is a question in the chat. Should I try and read that now, or should I wait until the end? Maybe I'll just wait until the end, unless it's something urgent, and then please just unmute yourself and speak up. So since in this paper they did analyze a topological transition for a Hermitian system, and we have a non-Hermitian system. So now we would like to interpolate between the two systems to see if this the physics still holds. And so what we do here is now to generalize our phase space by introducing transitions in the reverse direction. So before, let's say we had an arrow pointing in just one direction. Now here we have the reverse direction, and these two are equal in the Hermitian case, and they are not equal in the non-Hermitian case. And then we can write down, solve for the spectrum of this full model with now with all of these 4 different parameters. So the original parameters in and x, and then their reversed ones. And it looks something like so. And this is a little bit messy, so we'll take a moment to tidy up our notation by reducing 4 parameters now to just 2 main parameters that we can investigate systematically. One of the parameters that we'll choose is simply R, which typically in the Hermitian case tunes the system from between non-topological to topological. And so that is the ratio of these black to these gray bonds. And so when the transition happens at R is equal to half. And when R is more than half, then the external is stronger than the internal, and vice versa. And now this is new for our non-Hermitian system, this C. This is what interpolates between the Hermitian system at C is equal to half and the non-Hermitian at C is equal to 1 or 0. So the case that I was telling you earlier, with the chirality completely in one direction was C is equal to 1, and the system with the opposite chirality is C is equal to 0. And now we can see that the system remains topological as we change the chirality of the system from Hermitian to the fully chiral case. And so above a certain critical value, R star, the band gap remains open, and which shows that the system remains topological. And so we can see here that the band gap here remains open even as we sweep across above R star. And R star, as I mentioned, is half for the Hermitian case. And this is from this previous work. And also in our fully chiral case, we can show that it is half as well. And in between, however, we actually need stronger external transitions. So we actually have a higher value of R max. The maximum value is 0.69. So at any point, if R is above this R star max, then we can safely change chirality with the band gap remaining open. So now we can compare between the topological phase and a non-topological phase. So here this is R is more than half the topological phase on the right, and this is the trivial phase. And here I plotted periodic boundary conditions in gray, and we can cut the system open to look at any edge states, which is plotted in blue. And I've also further colored any states that remain only on the edge in red. And here what we see is that in the topological phase, we have 2 rings of edge states that emerge. And in fact, these 2 rings will then shrink towards, this ring shrinks towards this point, and this ring shrinks towards this point as we start to increase R even more and more towards going even deeper in the topological phase. And these points that these rings shrink towards are exceptional points, which are these non-Hermitian singularities that I mentioned in the introduction. And these exceptional points have interesting properties, one of which is that when you wind around an exceptional point, here you actually need to wind around twice to go back to yourself. And you can see that it's because here there are 2 different Riemann sheets that one can traverse before you go back to where you were initially. And there are also, there's also another nice, there's also another interesting invariance that was pointed out in this that can be present only in non-Hermitian systems. And this is a new topological invariance that can be defined just by looking at the eigenvalues of the system, which is here defined below. And this is nice because usually in Hermitian systems, we define topological invariance using the eigenvectors. And so while the Berry connection that I first mentioned, that was introduced by Zach in lattice systems, can be defined in both Hermitian and non-Hermitian systems, these quantities, instead exceptional points and vorticity, do not have any Hermitian analog. And what we can see is that when we just, as I was saying, if we increase R, so here this is a topological transition, this is just a real and imaginary spectrum. And what we see is that as R continues to increase, that all of the states start to get dragged to two particular points, which are exceptional points. And so that you can see that the states here coalesce to these exceptional points, which marks one of their properties. And we'll also see vorticity by investigating a half-periodic geometry. And here, in this half-periodic geometry, the system is periodic in the y direction, but open in the x direction. And so we can plot the band structure along the system, which still has the reciprocal vector. And what we see here is that as we tune the system from being fully Hermitian, so in this Hermitian regime, the system is fully real, it has no imaginary components. As we start to change it to become non-Hermitian, we start to see edge states emerge, which are these states which have the highest dispersion. And they also have an imaginary component. And what we find is that these edge states also have interesting properties, that initially that they are real and they are equally distributed on both ends of the system, of the open dimension. And as chirality increases, they start to develop imaginary components to become localized on only the left or the right system edge. So here you can see that they're red and they're distributed, equally distributed on both sides, and then they split. And then here now the green band would be distributed only on the left and the orange band would be distributed only on the right. And as we increase in non-hermiticity even further, at some point, actually, there's a qualitative transition in which now these bands merge and touch. And so then we start to see this winding complex space with a non-zero vorticity. And you can see this double periodicity that you have to go around the system twice in order to get back to itself. So now here we have seen what topology can tell us about stochastic systems. And then we can go back to thinking about what biology can introduce that's new. And so before I go on to this last part, are there any questions? Okay.
I have a question.
Yes.
Do you have just imaginary eigenvalues, not just exceptionally, just imaginary, not real part, or
Yes.
the symmetry breaking part? Because when we are having non-Hermitian systems, we have three kinds of things, like real one, exceptional, and breaking the symmetries. So do you have also the breaking the symmetries and just also the imaginary part, not the real part?
So we do have, so let me show it. So whether or not we have, so this is, yeah, so you can see that here the system is plotted in both real space and imaginary space. And you can see that there are states that are, well, no, so actually, yeah, interestingly for these master equations, no. If we were looking at electronic systems, that would be possible, but in these master equations systems, that's not possible. So the system here, it has always a real part, and then it has imaginary components here.
So you cannot tune the R in a way that it just have imaginary part, correct?
So you mean to have a zero real part?
Yes.
So no. So especially in one property of these stochastic systems governed by the master equation is that it has a unique degenerate state. And so as a unique degenerate state, that state is always at energy zero. And because it's unique, it cannot actually have two states. And so it cannot have an imaginary part. And this is part of the Perron-Frobenius theorem.
Thanks.
Sure. Okay, great. And so I will just finish up. There's just only a few more things. And so now let's think about how, what are some biological considerations that we can have, especially as some of the questions that already have been placed, which is, well, isn't biology even more complex than that? And so, yes, it is. And so let's look at how some of this can yield also interesting behavior. So here, this is easiest to illustrate in a 3-state model, which here there are these 3 internal states and that's placed in a larger triangular geometry. And so here, these coordinates could describe something like here, this could be addition of GTP bound monomers, converting them to a different kind of monomer, and then removing those converted monomers. And if we plot this as just this axis as the number of monomers in the system, because this is conversion, so there's only one type of monomer in the system. And we look at it, we see that the number of monomers grows here, it converts and it shrinks, it grows, converts, and it shrinks. And this is just as we expect. And here we have just 2 rates, the black and the gray. Now, of course, biology need not be so symmetric. And in fact, we can change just one of the rates. So let's say we increase one of them, we increase one of the gray rates, which is now the upper right rate here shown in blue, to become somewhere in between the black and the gray. So it's not as fast as the black, but it's also a little bit faster than the other gray rates. And so what happens is that here, down this edge will be a bit faster than these other 2. So the system will grow, will convert very quickly, and then it will shrink and grow, and this one will be fast, slow, slow. So now if we plot this monomer axis here, what we see now here is that this is very fast. So it grows and then it shrinks, and there's very little conversion time. But not only that, now because that this is a bit faster, sometimes the system doesn't make it all the way to the edge. Sometimes it unbinds halfway in between. And so that's why you see that there are these moments where it doesn't make it all the way. And so there is this stochastic unbinding. And this is somewhat reminiscent of this dynamical instability of microtubules, which has been observed that these are part of the cytoskeleton in the cell. And these grow and then they start to shrink, and then they grow and then they shrink, and when they start to shrink, seem to happen in an abrupt way, in an abrupt time. And of course, microtubules are very complicated biochemically, and this is a very coarse-grained system. But it's interesting that we can reproduce these complicated dynamics with just 3 parameters, which is the black, the gray, and then one of them that's tuned, just by repeating these motifs in a versatile way. And then the very last thing that I'll show is that now here, because we can associate the axis to adding of monomers, for example, so that so then if the monomers are obtained from the same pool in a system, then we could have more than one system that's obtaining monomers from the same pool. And then once they finish the number of monomers in a system, then they have to change in their reaction space, and then they will move up. And so then the boundary now is set by when they both deplete the number of monomers in the system. So now we plotted the system, one is growing this way and the other one is growing towards it. And once they hit each other, they've depleted the monomers, they have to convert. And so what is interesting is that after a while, because of this competition, you start to see that after all, they start to show this anti-synchronization, where one will sort of go all the way up first and then the other will go all the way up. And the other thing that is interesting is that you can see that they don't always all go up at the same place, that because it's a stochastic system, that this shared boundary that they have will keep moving. And this is also another interesting point that does not exist in solid state systems, where the boundaries of the system are fixed, whereas in this case, we have dynamical boundaries. And this was in the case in which we have, again, symmetric internal rates and just two types of rates, the black and the gray again, where we obtain anti-synchronization without directly coupling the systems and just by having shared physical constraints. And then, of course, there's a very large phase space that we can play with. Lots of different reactions could happen in here. So just by changing one or two of the rates now here, we see a regime that supports some in-phase synchronization. So this is what I wanted to tell you today. And I'm just going to finish by telling a little story, which is that in my very first paper, which I wrote with Xiaogang, who I think is here in the audience, we predicted a new platform for a topological phase with novel properties. And I even discussed this with Georg Tchaikovsky, saying that, well, it's a nice idea and it's probably pretty hard to make. And I was really happily surprised 7 years later when very heroically they synthesized this material by plunging powder into 750 degrees Celsius and then submerging crystals in an ice bath. And I think that this is, helps to illustrate why I remain fascinated with topological states after all of these years, because I can propose a theoretical model and then these states can be detected or synthesized years later in an unexpected way, or in many different new platforms, which is a lot of fun. And so I await good surprises, maybe also in biological systems. And with that, I would like to thank my collaborators. This work was mainly done with Jaime here, who really brought the knowledge of stochastic systems to the table, and with Ramin Golastanian, who is the director of my department. These are my students, Lorenzo and Lara, and Benoit is another collaborator, as well as Andre. And these are collaborators that from the University of Pennsylvania. And with that, thank you for all the great questions and for your attention.
Thank you very much, Evelyn. Do we have any questions? If somebody from the audience has a question, please just unmute yourself. The question in the chat was already answered, so you can ignore that.
I'm seeing the chat. So I think the chat was saying about whether we looked at the non-Hermitian Hamiltonian for the Berry phase. Yes, we did. And one can generalize the Berry phase to have a non-Hermitian description as well. So let me see if I have a slide on that. So here, this is typically what happens when you have a Hermitian Hamiltonian. And then if you have a non-Hermitian Hamiltonian, one would use both the left and the right eigenvectors of H. And so then integrating this is a complex quantity, but in the presence of symmetries, it's still quantized. And so, and these symmetries do exist in our system. Sorry. I was just trying to find my summary slide, but anyway, I think I have too many slides. Maybe it's this one. Okay, perfect. Are there any other questions?
I have a question. So in the systems that you've told us about so far, is it correct to say that they're free systems? And can you imagine in biological context if there is a way to where interactions are relevant and what the implications of that might be?
Yeah, that's a great point. I suppose, well, one question would be, well, what kind of interactions are present? And I think that the last example that I showed of synchronization is a question in which one can think of how these systems could be interacting and how one can obtain emergence dynamics in such systems. So I'm going to show that again. So here you can see that we can have them interact not by directly coupling them because oftentimes, one could write on a model in which one could just add interactions, but then the question is, well, microscopically, how would, let's say, monomers or polymers, what is actually the coupling mechanism between them? It's usually probably some kind of chemical reaction process. And so yes, of course, we can add different interactions, and that would be very interesting to consider. And I think that this is a place in which we are starting to consider that and to think about what kind of different emergent dynamics one could get from here.
Excuse me. Hello?
Thank you.
Yeah. Hello? Can you hear me? Yeah,
Hi.
I have a question, and
Yeah.
it's actually related. It's have you considered a noise? Usually, when I have a dissipated system, there will be a fluctuation, a noise.
Mm-hmm.
And it turns out that for this kind of a master equation, the noise actually correspond to interaction. When you include noise, that effectively including some kind of many-body interaction. So But generally in biology, there's a lot of noise. If you include noise, I wonder whether there's something different or similar thing happening.
Yeah, that's a great question. And well, I would say that there is a lot of noise on the biological level, but there's also a lot of very strongly directed reactions. So, for example, when ATP is consumed, then the system is very strongly biased in one direction. So that even if there is noise, the system, will still move in that direction. And the noise comes in that this is some kind of Poisson process or some kind of is better to model it using some kind of rate reaction. So that is one kind of noise that is built into these stochastic systems. And of course, there could be just fluctuations that are present. And I think that would be a different kind of noise that would be interesting to consider as well. Probably the strongest amount of dissipation comes from this sort of ATP consumption, but of course there is different kinds of other kinds of noise as well. And yeah, it would be very interesting to think about that more. Can we use ideas about robustness or about the spectra of the system to quantify how much noise the system can tolerate? I think that would be a very interesting direction. Is this what you were asking, or were you?
Similar. You consider the master equation, which is a differential equation for probabilities.
Yes.
But you could add a noise, make it a random differential equation. There's a random terms. And you can simulate this differential equation with the random terms and to see how the behavior of average is robust against those random terms. This master equation with a random term is a kind of random differential equation, which since maybe the simulation it's a little bit different. Yeah.
Yeah, that would be very interesting. And I think from the simulation point of view, like that's definitely something that could be done. Already it's easy to simulate these things. From the analytical point of view, I don't know right now whether things like the Berry connection, etc., could be generalized for systems like that.
For an analytical point of view, when you're adding noise, it's equivalent to adding many-body interactions.
Mm-hmm.
So it's like going to interacting system. Yeah, that's my comment.
Interesting. Yeah. It could be interesting to discuss this more.
Yeah.
And see how that could work. Thank you.
By the way, there's a path integral formulation of including noise. Then you can see that's indeed an interacting non-Hermitian interacting system.
I haven't looked at that yet, but that seems something that would be pretty straightforward to write down.
Yeah.
Hi, Evelyn. Can I ask a sort of a naive question here? Sure. Hi, good to see you. You have these edge states, but like in the condensed matter context, the edge states are not just directional, but also quantized. But I didn't understand what is the analog of quantization in your case? Like, is there an analog of quantized conduction or something like that?
I don't think that there is, at least, not as far as I can see here. Definitely, the amount of probability in a system is quantized, and that's because we have, let's say in this case, the entire system is moving along the space. So, if you start to stack many systems, and then you can start to see multiple copies of this. But right now, this would be equivalent to, one electron moving around. I would say that, to think about what is the kind of protection here that you see really comes a bit more from this exponential localization on the edges, and that is where I think we can see that the edge state remains there and doesn't penetrate into the bulk. So that is one way, a different way of seeing the topological protection, I think, in these systems.
Okay, thanks.
Sure.
Okay, if there are no more questions, then I think we're more or less out of time. Thank you very much, Evelyn, again. It was a great talk.
Thank you. It's
Yeah,
really,
thank you
really
very much. Nice talk. Thank you.
Thank you.
Thank you.